Shall I start now?
Let me just, I usually study like 17, so give
Okay.
1 minute, yeah. Okay, right, yeah, we go ahead and get started. I guess it's not, it's a little bit too late, but still say Happy New Year to everyone. This is our first grand round for the new year 2023, and welcome everyone. Now, for today's speaker, I do not need much more for the introduction. Peining Li has been a long-standing member of our faculty and also the director for Cytogenomic lab. But I do want to kind of emphasize his expertise in this particular area for understanding cytogenetic abnormality from the pregnancy loss, which is he did extensive work with Alan, Wei, and Chen, and other people in both DNA and Cyto lab, led to a successful grant. Right now we're running here for the recombinant pregnancy loss from NIH, which is one of the major sort of project in this direction. So, so with that, I will let Penny take the floor.
Okay, everyone can hear me, right?
Yeah.
Okay, yeah, thanks for the introduction. Actually, today's talk will more focus on the cytogenomic part, but that means the chromosome abnormality and the copy number variants. And then the gene part is kind of under a research project, so I'm not going to touch that much into that. Definitely, Zhao Chen and Hui and Alan, we already published that the preliminary study on that direction. So today's learning objective is just give a current understanding on the spectrum of cytogenomic abnormalities in POC. You know, I kind of do a meta-analysis on 7 large case series by microarray and chromosome analysis. So that pretty much give us a overall picture about how is the cytogenomic abnormality affecting the pregnancies. And then moving forward, I will talking about how to estimate the risk of spontaneous abortions caused by mainly the major pathogenic copy number variants that's to the genomic disorder and syndromic pathogenic copy number variants. And then toward the end, I will try to touch the functional implication from cytogenomic abnormalities. So as we know, actually, a pregnancy will take about 10 months. So usually this been divided into 3 phases: first trimester, and second trimester, and third trimester. And then they come from the conception toward in the first trimester will come from conception to implantation. But during that period of time, they will experience some like early miscarriage or late miscarriage in the first or second trimesters. And then this condition actually is quite common. And then there's one estimation based on the published book. It's like if you have come to clinical recognition of a pregnancy, usually this is on the top part of this. And it's like probably 1/4 of that will be lost. That means the pregnancy loss. And then only 3/4 of those will survive to term. But if you take all the conception under consideration, probably 60% of loss or failures is not even recognized, not even bring to clinical attention. But usually we'll take this top part, like how to estimate the risk of spontaneous abortion. If you follow this, usually the risk is like 25%. But in textbook, generally, we take 50% as the baseline risk for pregnancy loss. Now, this shows the phases for fetal development. So usually it goes through all the first 10 to 40 weeks. And then the first 10 weeks usually is the early embryogenesis and organogenesis. And then toward the second and the third phase is getting more into the functional part. So I think this is a pretty basic for fetal development. And then there's a big debate in like in United States, like which stage is considered you can do clinical abortion, like which stage you will really consider to be alive. So this have not only just the medical termination, but also have legal relations to that. So this is how the early stages in the first 40 weeks for life. But once we get into this specific terms for how we describe early fetal demise. So usually the terminology is based on the 2003 revision of the ICD-10, and loss of a fetal at any stage is considered a fetal demise. But if the fetal demise is less than 20 weeks, and it is usually classified as spontaneous abortion or miscarriage. If it's larger than 20 weeks, it's usually classified as stillbirth. And in clinical practice, laboratory usually receive the so-called products of conceptions. That's come to the title of this talk. Is like this is tend to use to identify any tissues that develop from pregnancy. It's usually to refer by doctor to collect any tissues, and then for POC samples, we usually can receive placenta tissues, or core skins, or sometimes even the whole fetus. I mean, for people in the lab, they process this tissue. They usually will get the villi tissue because this tissue is considered the most successful in cell culture, has 90% of cultures. But if you go to other tissues, the culture success rate decrease. So that's the reason cell culture failure is a affecting factor for POC analysis. And in lab practice, we usually will have a POC specimen, and then based on what I'm saying, we need to set up for cytolab, we need cell culture, and then karyotype it. But we encounter a significant portion of those, 10 to 20% will be cell culture failure. And then early days, usually we don't generate any report for culture failure samples. But once we introduce the microarray technology, actually we can extract the DNA from the culture failure and then still do the genomic analysis. And then there's a significant portion of those will end up like normal karyotype. So the normal karyotype, you also can do DNA extraction and then do the microarray. And then generally, the chromosome abnormality is seen in 50% of the culture success cases. And then once we go through the microarray analysis for culture failure, normal karyotyping, and we detect the chromosome abnormality in 35% of those culture failures, and then we detect pathogenic copy number variant in 4%. And then this table actually summarizes some of the data in 2016 from Cytolab. And then this is a table shows the chromosome abnormality in karyotypings. This is over 1,000 cases. And then we also do additional microarray analysis in culture failure cases. So this will be usually the spectrum of the chromosome abnormality identified in POC. And then once we introduce the microarray, and then we encounter a request from doctor to say they do have tissues, paraffin fixed tissues from POC. They want to get into the information about the chromosome abnormality. So we also develop using the OncoScan microarray. This is a SNP-based microarray, and it's a specific design for kind of a fixed or broken degraded tissues. So actually, we introduced that technology for PCR FFPE, and then successfully show actually we can detect the similar spectrum of chromosome abnormality and additionally pathogenic copy number variants. Now this is pretty routine right now in the cytogenetic lab for POC specimens. Now for the diagnostic yield of chromosome abnormality, we can see actually there's a most frequently see chromosome abnormalities, so trisomy 13, 15, 16, 18, 21, 22, and also monosomy X. they all account to larger than 5% as a relative frequency, like counting toward all the abnormalities. So to the scenario of typical POC, and culture failures, and even in special scenarios like multiple anomalies, we kind of give this distributions here. There's one hypothesis, I think, in the early early 2020. So like they kind of the hypothesis to say that culture failure cases will encounter more like a monosomy, because they are monosomy, they are not that easy to success in the culture. But we put the monosomy cases in here, you can see actually most is actually from the culture failures. And the other part, monosomy 21 is also see in POC, but those are very real scenarios. We still don't have enough case number to evaluate how impact from the monosomy part. But another thinking is like toward those most frequently see trisomies, there's also another hypothesis, and maybe the less gene number in those chromosomes make it see more frequently. So we also plot this relative frequency toward the number of genes in each chromosome. And then this 6 chromosomes outstanding are actually those most frequently see. They have some kind of gene number related to it, but we still need to dig out more to see how this trisomies or monosomy will affect abortions. So this is the summary of the pathogenic copy number variance. And then out of 435 cases, we detect about 4% have pathogenic copy number variance. Further narrow down this to the syndromic or the genomic disorder, that means the microdeletion, microduplications. Actually, this will down to like 1.4%. I highlight here the right ones are for the syndromic and genomic disorders. So this is the study we kind of summarized in 2016, and to give further understanding on the, especially on the pathogenic copy number variants and also the genomic disorders. And then we did a kind of a literature review for large case series, because the percentage of the pathogenic copy number variants is kind of low, like a 2 to 4%. So we really want to get the real diagnostic yield and the spectrum of the pathogenic copy number variant, especially the genomic disorders in POCs. So we kind of review about 7 large case series. When I say large case series, the case number has to be more than 1,000 cases. And because the case less than that is kind of introduce a lot of barrier. So this all down by microarray analysis, and then the case number range from 1,000 to 25,000. So, and then the diagnostic yield for chromosome abnormality, pathogenic copy number variant, and genomic disorder was summarized here. And then to give an estimation about the diagnostic yield, and then we did actually we have a Peng Gang and from Peng Yu from public health, and then to help us to do a meta-analysis, so to give a real estimation and then show the variation from different studies. So for the chromosome abnormality, based on the common effect, that means counting the case number with some impact. And then the estimation for the diagnostic yield is 49.9%. Actually, that's what the already known in the past practices, like a 50% will have chromosome abnormality. So this is the statistics data. And then once we counting toward the general pathogenic copy number variant and more toward the genomic disorders, and then it's about 2.5% have the pathogenic copy number variant, and then it's about 0.8% will have the genomic disorder and syndromic, more severe pathogenic copy number variants. Out of these 7 studies, and then we see like there's one study for the pathogenic copy number variant, and one study for the genomic disorder is kind of outliers. And then when we look into the data, so actually the way they classify pathogenic copy number variant and the genomic disorder probably contribute to this kind of outlier. And this also bring out the concept of how we're going to define this. The general practice for pathogenic copy number variance is like a less than 10 megabase will fall into the pathogenic copy number variance. And then genomic disorder and syndromic ones, so there's a little debate in there. Genomic disorder is pretty straightforward. The recurrence of genomic disorder in population is based on the low copy repeat in our genomes. So there's a genomic structure predisposing risk for microdeletion, microduplication syndromes. But the syndromic pathogenic copy number variance is falling back to like the early days. We using FISH to detect subtelomeric deletion and duplications. So that actually is getting into syndromic by sharing critical gene, critical regions. So this is pretty much based on this meta-analysis, we get a whole picture of like what proportion of those patients, those POCs, will have chromosome abnormality, what portion will have pathogenic copy number variance. And the genomic disorder and syndromic pathogenic actually is falling into the pathogenic copy number variant, but it's accounted for about 1/4 of those if you count from 2.9% to 0.8%. So this is the overall picture for cytogenomic abnormalities in POC. But in the current studies, because we try to kind of do the genomic analysis for the recurrent pregnancy loss. So this definition about recurrent pregnancy losses, like if you have 2 or more pregnant losses, they will consider RPL. And then usually in diagnostic practice, that's kind of labeled in the gray zone, is because any case referred to a cyto lab to do chromosome analysis is not the first pregnancy loss. Usually, once you have the pregnant loss, the doctor will say, this probably happened. And then until it's the second time, and then sometimes even the third time, you'll go for the cytogenetic analysis to find out the genetic causes. So actually, if you follow, more stringent, the recurrent pregnant losses, this is probably, it's very difficult to define. From the lab point, but actually we review our database and then to find those cases with a clear clinical indication of recurrent pregnancy. That means multiple miscarriage and recurrent pregnancy. And then we find out of our over 4,000 cases for the past 15 years, it's only 6% have a clear clinical indication of pregnant loss. And then from this group of patients, we have 18% culture failure. So it's about 23 some cases we do the chromosome analysis. And from this group, actually, I divide this into 2 analyses. One I call a single analysis. That means we only do one time for that patients, even though the clinical indication have multiple miscarriage, but we only catch in one POC. And then also looking into the cases actually have a consecutive analysis. That means during the pregnant loss, we perform in 2 analyses. So just briefly show the abnormal patterns. So quite similar to general POC analysis. So we can detect the simple aneuploidy. This is from the single analysis. So we can detect all the aneuploidies and some compound aneuploidies, polyploidies. And then because they are kind of recurrent pregnant loss, we see some of the translocation related. So that's ribosome translocation. That's a known factor to introduce abortion, also unbalanced arrangements. And then we also detect some pathogenic copy number variants. But when we put it into this category of a compound aneuploidies, this probably have a cause by some genomic variants affecting chromosome segregation. So because they affecting multiple chromosomes. And then for consecutive analysis, we perform on 9 patients, 9 pregnant women. So there are 2 pregnancies, we actually do the chromosome analysis. And we see some cases that have normal findings, and then some have like 1 normal, 1 abnormal. There's a cases we see actually identical abnormality. So like in these patients, 2 of the pregnancies show trisomy 16, and then in another patient showed trisomy 22. And then they have patients that show different aneuploidies. So this actually bring up to some kind of further follow-up studies, like if there's identical abnormality in consecutive analysis, there may be a concern for the maternal germline mosaicism. So this how to detect this, this is pretty challenging because you need to get the germline tissues. And then if you see recurrent abnormalities or even the compound aneuploid, like previous slide, and then they may be caused by pathogenic variant affecting chromosome segregations. So actually this probably, this kind of, if we have more study on this kind of patients probably need to do some kind of a further follow-up study in the family and also follow-up study on molecular mechanisms. So now I'm going to move to the second objective, is to estimate the risk of spontaneous abortion caused by genomic disorder and syndromic pathogenic copy number variants. So this actually is a lesson learned from the chromosome abnormalities. This table actually is adopted from the genomic medicine. This kind of well-established analysis for risk of spontaneous abortion for chromosome abnormalities. So this is based on theoretical, like 10,000 conceptions. So if you have 10,000 conception and then based on this abortion rate of 15%, this rate could be vary from 10 to 25%. But usually in this book, they take like 50%, probably a little bit underestimated. And if you have a 10,000 conception with abortion rate 15%, so you end up like 1,500 with spontaneous abortion, and then 8,500 moving to a live birth. And then we know there's a 50% chance they will carry a chromosome abnormality, so that will give us 750 cases. And then the other 750 will be chromosome normal. So based on this, there's estimation, if you're abnormal chromosome, 90% chance you end in a pregnancy loss, and then only 6% chance you will survive to term. By prenatal analysis with normal chromosome, it's only 8% chance you will have a spontaneous abortion, but 92% chance you'll move to a live birth. Now, if you counting specific type of chromosome abnormalities, triploidy and tetraploidy, 100% will stop in the prenatal, and then 45X monosomy X, 99% will end it, and then only like 1% chance they survive to term. So that also contribute to the instance in live births is like 1 out of 8,500. And then trisomy 16, trisomy 18 is pretty tough. There's a strong against those. So that will be 95 to 80%, 100% will be aborted. And then trisomy 21 Down syndrome, actually the spontaneous abortion risk is 78%. So that's what the reason we see some of those cases in the live form. And then the other trisomies have risk too. But for the sex chromosome aneuploidy, like Klinefelter syndrome and other aneuploidies, it's only 21%. So that pretty much close to the baseline kind of a risk. So there's not that much increased risk for sex chromosome aneuploidies. And then for unbalanced arrangement, the structural arrangements, if you are unbalanced, you have 85% risk. But for balanced arrangement, that means there's not a copy number loss. And then the risk is only 16%. So actually that's pretty close to the baseline risk. And then that means this most of the chromosome abnormality will have a high risk of spontaneous abortion. So we usually consider if it's larger than 90%, there could be a strong selection against this. But if like 70% to 89%, there could be intermediate risk. And then if less than 69% is considered weak selection against that. So there's also another message from the chromosome abnormalities, like there's a significant higher instance of showing this abnormality in POC than in lab births. If you do that, that actually is statistically significant. But now that bring out the questions like what's the risk for POCs, the pathogenic copy number variance in POCs, and what's the percentage they will be actually selected against the, through spontaneous abortions. We know actually that will fall into this category because you have normal chromosome, and then if you detect a pathogenic copy number variance, what portion of those will be aborted out. So that's what we try to find out this information, and then also from the large case series. So from that practice, we actually can give some estimation. This is a paper published 2 or 3 years ago based on the 10 years pediatric data. This estimation for all chromosome varieties from genomic medicine, this is pretty established. It's a 0.67% newborns will have a chromosome abnormality. That's what we see, and also in the POC we see it's like 50%. But once you get in, because 94% have been aborted, and then once they come to life, it's only 0.67%. And we also did an estimation from our 10-year data from pediatrics. We give an estimation probably like it's 0.34% will have a pathogenic copy number variant. So taking these two together, it's about 1%. So about 1% of newborns will have some kind of a chromosome copy number abnormalities. But the issue right now is like this estimation was done some time ago, because right now the prenatal detection for chromosome abnormalities is quite effective by the introduction of non-invasive prenatal testing. So we expect this percentage should be decreased, but this decrease probably will be more obvious like for Down syndromes, because we can detect Down syndrome more than 95% of those prenatally. So actually there's an ongoing study to see how our current practice will reduce those abnormalities in pediatric or in newborns. And also we can estimate how much risk for pathogenic copy number will have some abortions. So to do this kind of a risk analysis, we need to establish the instance of this pathogenic copy number variance in POCs. And then this will be pretty straightforward, because you collect a large case series. From that 7 large case series, you have 35,000 cases, POC cases. And then you base on the real number, and then you can give an instance here. So the instance, for example, for DiGeorge syndrome, the instance we estimate is 1 out of 1,000. And then DiGeorge syndrome in general population by population genetic studies is in the range of 1 out of 4,000 to 1 out of 6,000. So actually you can see in the POC, the instance is significantly higher than DiGeorge than the newborn. And then this highlighted here is the syndromic pathogenic copy number with known instance based on population study. And most of the other copy number variants, we don't have a population data. So we pretty much based on the largest pediatric patients, and then using the known population instance, and then to infer the instance in newborns. So based on the 4 known population instance, and then we getting the estimation like probably this kind of a patients we see is from a newborn population of 700 to 4,000 kind of newborns to give this, and then using this number, and then we can estimate the instance in newborns for all this specific genomic disorder or syndromic pathogenic copy number variants. So this list of genomic disorders is kind of we give this instance in both in POC and in newborn. And also you can see from the p-value to compare their instance, most of those are significant, but they do have a few of them it's not significant. So their p-value is larger than 0.5. So that actually bring up question, they may not be that much a strong selection against this kind of genomic disorders. Now this we do a pretty similar to the genetic in medicine, genetic in medicine tables. So at this time we based on if you look in the table I showed before, it's based on 10,000, but we increase like to 100,000 because the frequency, the instance of pathogenic copy number and genomic disorder is kind of low. So when we increase about 10 times, we can expect about 50% of those will have a spontaneous abortion, and then that risk, general risk, is about 15%, and then live birth is 85,000. And then we plot the instance in POC and then the instance in live birth, and then that can give a risk estimation by usually pretty straightforward. It's just like you add in you estimate the risk by divide the POC number to the two number combined. So like the 42% risk for DiGeorge syndrome is because we see 36 out of 57 cases through the estimation of the conception. Let me see, I make it right. So through the conception, we see 36 cases, but we aborted 15 cases, and then you divide the 15 by 36, that gives 42%. So this table actually is just follow the chromosome abnormality. We give an estimation for the risk of spontaneous abortion for those syndromic ones and genomic disorders. And then if you taking all this together, the estimated abortion risk is about 38%. If you compare the previous table for chromosome abnormality, that's about 94%. So this is a significant reduced risk for spontaneous abortion for this copy number variance. And then the highest one we see is probably in the range of 50%. And then there's a 50, 60%. And then there's some cases actually is kind of falling into like a 20, 25%. This is pretty much falling into almost toward the baseline risk for spontaneous abortion. And so we kind of try to introduce a classification for risk of spontaneous abortions. And based on the chromosome and genomic disorders, and then we kind of try to say like if it's larger than 75%, this consider a high risk for spontaneous abortion. And if it's like in the middle of this 50 to 75% is considered intermediate risk. And if 26 to 50%, that's considered probably low risk. But if you below 25, from 0 to 25%, because that's pretty much falling into the 15% range. And as I showed in the first slide, usually they could be 25% will end up a spontaneous abortion. So actually, this is considered probably baseline. And then if you look into that, we can see we plot some typical abnormalities. So like triploides will definitely, they will be aborted. Turner syndrome have a very high risk of 99%, Down syndrome 78%. And then there's not that many one falling into the intermediate risk, but like for DiGeorge syndromes is actually with the intermediate risk. And some of the syndromes actually like Williams syndromes, Klinefelter, Robertsonian translocation, these are considered baselines. So actually with this classification for risks, actually that can actually provide more information for patients in more diagnostic practice. So this is the part actually we try to figure out the risk for both chromosome and copy number variants. And then further down that, we'll try to see how why is the mechanisms relate to those kind of risks. And toward that part actually we'll bring out like three approaches. One is kind of a case-oriented approach, just like if we identify some significant case, we try to figure out the mechanism. And then the other is maybe some bioinformatic approach. And then further down will be like more like a function analysis to figure out the genes. So for the chromosome and copy number variants, so we're usually dealing with dosage sensitive mechanisms. So that probably is different from gene function, other Mendelian inheritance. So the dosage sensitive mechanism usually is considered haploinsufficiency. That means you lost a copy, they have an impact. And if you have 3 copy, they call triple sensitive. And then this is the 2 kind of mechanism we generally say that. And then if one gene have shown both hyperinsufficient and triple sensitive, that consider actually is a true dosage sensitive gene. But if only affect one, still consider have impact. But this area is recently, because of the copy number study by microarrays, is more extensively in clinical now. So we do have some data from ClinGen and then to figure out the dosage sensitive genes. So I want to bring like one case. We say like this. This is a micro duplication involve 15q13.1. And then the candidate gene is ChRNA7. Based on our estimation, this gene actually have a 40% of risk for spontaneous abortion. So when we look into this gene, actually, this is the old man gene. And if you based on the ClinGen, they give this hyperinsufficiency index, they give like a 40%. Usually less than 10% is considered hypoinsufficient. So actually, this is not a hypoinsufficient gene, at least up to today's data. And then another number they're giving is like intolerance for variant loss. So this was 0.77. Usually less than 30.35 is considered intolerance to variant loss. So from both this category doesn't show this gene is kind of functioning in dosage sensitive way, but the study I mentioned later actually indicate this probably is a candidate gene for pregnancy loss. So when we look in the function, this ChRNA7 gene coding a nicotinic acetylcholine receptor alpha-7 subunit, which play a role in kind of in 2 pathways. One is in calcium activated signal pathway, and the other is the cholinergic anti-inflammatory pathway. So there's a basic study using induced pluripotent stem cells from patients, and then they find actually this unexpected downregulation of this calcium signal cascades in both deletion and duplication. So this is kind of like bring out an issue, like you could have a deletion, you have duplication, but the downstream impact is all kind of the same direction. It's down regulations. So this is kind of an interesting finding from basic research. But the function wise, actually increased level, if you have a duplication in this case, and then it's correlated with excessive inflammation and TNF synthesis, they cause morbidity and mortality in diverse human disease. And so I think probably this dysregulation of these 2 pathways modulated by ChR may relate to a spontaneous abortion. So this is from kind of a specific deletion duplication, how to figure out their function impact on spontaneous abortion. And also in 2017, there's a study by Baylor's group, and then they also collect the pathogenic copy number variants in the pregnancy losses, and then do a kind of a bioinformatics analysis, try to figure out the essential developmental genes. And then they pretty much figure out some of the related pathogenic pathways involved in embryonic development, neural development, and also gene transcription and regulations. And then they kind of also given some kind of network, gene function networks. And I bring out here is a few of those actually relate to apoptosis. And so this pretty much, they summarize their data to say they probably, from pathogenic copy number variants, they predict about their 700, over 700 developmental genes mapped to chromosome regions may contain genes that relate to kind of spontaneous abortions. but this will depending on further analysis to see is this gene working in certain way to cause the spontaneous abortion. And then I want to kind of end up in one of the efforts from Cytolabs where we try to do kind of based on the aneuploidy to try to see if we can find the triple sensitive genes in common aneuploidies. And as I mentioned, like we do see this in quite common in abortions in POCs. So this account for about 60% of the chromosome abnormalities. And then we try to collect those POC sample with this common aneuploidy and triploidies. And then we kind of do it in a gender matched way. And then to do RNA sequencing and small RNA sequencing. And then if we find something, we try to confirm that currently by a merfish technology, they can do multiple genes. And then in that way, we try to identify the triple sensitive genes in individual chromosomes, and also try to see that we can find a way and interactions between these chromosomes, and then to find this any shared pathways relate to abortion. So this is a work done for the past 3-4 years and by Hong Yan, and then he kind of goes through the POC clinical data, and also collect the residual material, and then culture the cells, and then do the RNA extraction, and then submit to YCGA to do RNA sequencing. We try to kind of collect enough, but still a few abnormalities we still not collecting enough for those. Like for male trisomy 16, we only have 3 cases, and then for male, even female trisomy 18 and male trisomy 21, we haven't collected enough cases to do analysis. We try to get in at least 4 cases for both genders, and then we'll do gender match expression comparison to see any gene expression significant change. So we have some preliminary data, so I'm just briefly go through it. See, I cannot move the slide. Okay, so as we know, like a triploidy, every chromosome 3 copy, that's kind of high risk, 100% cannot survive to term. And then this is the RNA sequence data. Usually we kind of get into find the gene up expressed or down expressed. And then this is the statistics to see the expression level. And then this is the number of gene up and down. If we're looking for the up genes, it's kind of a little surprise to me. It's like only 83 gene show some overexpressed, and then kind of similar to the gene down. So by triploidy, and we kind of facing a situation if every copy is down, there's not that many gene actually overexpressed. And then the male triploidy, it seems like a more gene involved, but actually still, compared with the 20,000 human genomes, that means the overexpression gene or down-expression gene is kind of limited. So moving to a specific chromosome, if we're looking for Turner syndromes 45X, and then we can see this is the down gene and up gene. So it's only like 138. If you're looking specifically for, this is a chromosome X, and then this right spots actually represent the genes in X chromosome being upregulated, upexpressed. And then moving to like a trisomy 16 is also, this is the numbers, it kind of surprised it's more than the triploidy. And then bring to chromosome 16, it seems like they have more gene like overexpressed. And then this is, you can see the expression level for 16 actually is significantly show up expression. So this is actually probably explain why trisomy 16 was strangely selected. There's 100% aborted, there's no survivor to term. Trisomy 18, if you look in the expression, actually, trisomy 18 is this overexpression scenario. So this red spot gene is also is the one we consider probably triple sensitive. And then overall from the genome, affecting about 180 genes for up expressions. And then trisomy 21, you can see chromosome specifically 21 is expression is increased, but the significant one gene, the genes is quite limited. It's only like a limited number of genes. Total genome is like 42 versus 33. So this is the trisomy 13 similar scenarios. There's chromosome 16 gene is overexpressed, but significant ones, there's those red dot here. So this is for both female and male. We have a matched case to do the comparison. And then trisomy 15, similar story. We have kind of overexpressed the genes. And then we see more gene overexpressed in 15. And then trisomy 22 is kind of a similar story. And we kind of, if we getting into the whole gene number, putting into each individual chromosome. So we call this a dosage effect gene in common aneuploidies. So if you see Turner syndromes, actually we don't see that many genes in chromosome affected. It's quite limited. But it's kind of equal down and up. And then probably, we need to dig out more because Turner syndrome have a high risk of spontaneous abortion, about 99% of those. And then trisomy 16, if you look in genes in chromosome 13, the down expressed ones are very limited, but most 37 of the genes considered significant overexpressed. This is for both female and male. In males, it's 23 genes. And then trisomy 15, both male and female have significant number of genes overexpressed. Downregulated, down expressed one is very limited. So this is the 15. And then 16, kind of a similar. So, and then 18, we have 30 genes up. And trisomy 21 is kind of only 7 genes. And then 22 is in the gene of 60s. So actually right now we getting the information, probably from chromosome level, it's a little underestimated than I thought, but actually those are the numbers showing the up expression one is considered candidate genes for the triple sensitive genes. We need to do a little bit further analysis to verify that. And also we try to kind of do some, because they're also affecting our genes in other chromosomes. Want to see is there any common genes kind of could be affecting a shared pathway or something. So this is a kind of from trisomy 13, 15, and trisomy 22, we kind of list the genes. They are overexpressed in both male and females. So those are the 12 genes in the trisomy 13 scenario. And then trisomy 15, 47 genes, trisomy 22, we see 46 genes. This gender, don't remove the gender and then see the shared upregulated genes. It's not limited to that chromosome. So like for this gene, like NTM gene affected in both, in all 3 scenarios, this gene is in chromosome 11. So right now we want to do further analysis, try to see is there any, not only the gene in that trisomy chromosomes, and also probably the interactions from that gene to other chromosomes, and then to see is there any shared pathway involved in spontaneous abortions. So also there's like a llama genes and then LLPA genes also involved in 2 of this. So there's a lot of pending studies and for RNA sequence data. And also for the gene regulation part, we also have microRNA sequence data. We haven't do any analysis yet. So I think to end this, I think we need to find out the genetic etiologies. Right now, we from clinical diagnosis, and we pretty much know like chromosome abnormality cause 50% of the abortions. And pathogenic copy number and genomic disorder probably 2.5%, may vary from 2 to 4%. And then there's other causes of Mendelian genes with pathogenic copy or epigenetic mitochondrial genomes. So actually they need to do a lot of more genomic and genetic molecular studies to find out those. And then Yonghui mentioned actually the project currently ongoing is actually falling into this NIH pregnancy loss studies. So the concept behind that is they introduce a so-called interrome. And then the interrome is defined as a gene and chromosome regions critical for human development, the loss of which is incompatible to survival to birth. So this actually considers essential critical genes in our genomes. So, this part of the study, I think is ongoing. And then for my lab, and then we actually put in a little effort to look into dosage sensitive mechanisms for related to spontaneous abortion. So I think that's pretty much for this round. And then thanks. And then we also need to understand, the 10 months during pregnancy, actually, there's a lot of genetic things behind that. And there's a lot of study need to figure out what's the mechanism. And some of those actually is considered good, like it seems like 94% of chromosome abnormality being eliminated to birth because of the spontaneous abortion. But probably there's more to understand why this happened in humans. Actually, human as a species has probably is the highest spontaneous abortion rate compared with other animals. Okay, so that's the end for this, and then I'm open for any questions.
Thanks, Penny, for great summary and very insightful analysis. Yeah, let's open a question, comments for anyone. You can just unmute yourself, and or if you want to type in, that's also fine. Just remind you the CME code, it's in the chat. So if you need a CME code, please do so.
Yonghui, as I mentioned, like the RNA sequence data, we'll try to also do, collect some more cases and then kind of serve as a pilot study to how to use an RNA sequence to figure out the function for abortion-related mechanisms. So
that's quite interesting. I say also intriguing for particular for tri-plurality. It's so small number. You're right. It's so small number.
Yeah, kind of a little bit surprise to me when I see that number, and that just means a triple sensitive gene probably need some more focused study to figure out, and then why they have this. And when we kind of look into this, is like we already taking into the consideration there may be some regulatory issues. So the microRNA part could also be interesting. Yeah. Hi.
Go ahead. Yeah.
Yeah, hi, this is Dr. Bastar. I'm working at Yale Fertility Center. This was a really great presentation. Thanks very much. So I would like to, really curious about your opinion about, like, there are some publications that that they say that the embryo can cure itself even if it's aneuploid. What do you think about that one?
The embryo can what? Can
cure or make it correct the aneuploidies. Well, what do you think about that?
I think that's falling into the category. We in prenatal testing, we encounter scenarios confined placenta mosaicism. So that means if in the conception, the early days, the early 1 or 2 weeks, there's a lot of, probably just early 1 or 2 days, there's a lot of mitosis going on. And then anything that happen with errors in chromosome segregation end up like a trisomy or whatever. And then they will move that to a placenta. And then that way, actually, the fetus, the conception grow to the embryo still normal. This is a pretty common observation in early studies. And recently, actually, from the IVF and practice, and also from the clinical, especially in some like publications, they kind of take the risk to say if you detect a mosaic case, you can still let a baby go to term, and then when they come out and then they've been self-cured. I mean, this is they do have publication on that part, but I'm a little cautious of that because this is kind of without kind of a large case rate to verify that, and without further understanding why they can correct to the level like complete health, you still end up like some kind of a germline mosaicism, you know, undetected to the, you know, in the fetus. So it should be, it's a sensitive issue, especially in diagnosis for mosaic cases. And some practice, I think some clinician hear a seminar and see their publication in Taiwan, they kind of take a risk to convince the pregnant woman, oh, you have a mosaic trisomy, you know, just go ahead and then they will be self-cured. They do have a publication, but you know, this is not still pretty limited. Need to accumulate more
Exactly.
evidence.
Yeah, I'm so sorry. We had a, you know, was that professor here? So he had a publication too. He just transferred totally unapplied embryos, and he said that you know, pregnancy rate is almost 50%. And what was that? And miscarriage rate is almost 25%. I know, but it's really, you know, really hard decision to give. Thanks very much for the
I think
again.
In IVF, actually, they do have this. I think in there's one report from New York, and they end up like they see they do the IVF testing, and then they find the few embryo they select all have trisomy, and then they take the risk to implant that and then end up like a healthy baby. They do have this kind of report, but how much a risk the pregnant woman can take it, that will be, you know, different. And that's an interesting phenomenon. And then the self-correction is also, they have some study on that. You know, the cell can actually correction to a certain way, and once they correct it, and then they end up like some kind of mosaic situation. And how to define like this cell is a self-correction, and then the cell is kind of constitutional mosaic. They need some test because the self-correction one can end up something like a uniparent disomy, isodisomy scenario. So they need some molecular study and then to see how effective the correction of those. So
do you know any difference between the somatic cells and the, you know, the embryonic cells about the self-correction? I'm just curious about it. I never checked that one, so.
I don't remember anything, you know, immediately, and they do have some study in cell lines, patient-derived cell lines. They observe the corrections, and one of the major areas in ring chromosomes, they see that once they introduce patient-derived ring chromosome into induced pluripotent stem cells, and then they see the ring was disappear, be corrected by a normal chromosome. So this do happen in study, and then in real life, we need to further analysis, as I mentioned, to differentiate the correction one to detect the isodisomy to compare with the mosaic, the true heterozygous scenarios. In that way, we can figure out, oh, there's a correction they have. So they need the further molecular analysis to even to figure out those. Yeah.
We can take one more question from Tamo.
Hi, Peining. Great summary. Quick question. It's a bit technical one. So I am just wondering, the low number of differential expressed gene you get is somehow because of like low power. Like I'm just wondering, like, what is the number of samples you were comparing? Like,
this is a preliminary data. I think that the number is I show the number is like at least we have 4 cases, and we kind of you know, this kind of, even though they are commonly seen in POC, to correct a good number of cases is still pretty challenging. I think we perform about 600 cases if you POC, and then on annual basis, you only have 300. And then a good case can allow you to do further research is kind of limited. And then we try to do like 4 to 6 cases per gender for each abnormality. But if we can collect more, we can do more.
Okay.
But I think statistically, based on the preliminary data, and then 4 to 6 cases is sufficient to get.
Yeah, that should be good enough. Yeah. Thank you.
Yeah.
Great. Thank you, Penny, again. Thank you, everyone joined. Now it's about time. We're going to just finish here. Thank you. We'll see you next week.